Good afternoon, ladies and gentlemen, to this afternoon session on environmental risk assessment. My name is Juliane Kleiner. I'm heading the Risk Assessment and Scientific Assistance Department at EFSA, and I have the great pleasure and honor to chair the meeting together with Katharina Gansleben from the European Environment Agency based in Stockholm. We had quite fruitful discussion this morning about opportunities and challenges for applying ecosystem services in environmental risk assessment, and we also, I think we all agreed that we should have clear operational goals first and then work it back to meaningful endpoint. This afternoon we will dedicate to the diverse area of products on the horizons, ranging from gene-drive modified mosquitoes to RNA interference-based genetically modified plants and RNAi pesticides, systemic insecticide, genetically modified fish, you name it, biological control agents, and nanoparticles. It will be explored whether these emerging technologies and associated products may raise challenges for the environmental risk assessment and would benefit from new approaches to overcome them. We have a quite tight program. We have 8 speakers which will show with case studies how problem formulation can be used to formally draw plausible pathways, how a proposed activity could be harmful, to formulate risk hypotheses about the likelihood and the severity of such events, the type of information that would be useful to test and corroborate the risk hypothesis, and to characterize the risk, how this information can be guarded, and what are the underlying uncertainties. We heard this morning, as well as yesterday at the plenary session, problem formulation is often facing the absence of clear and operational policy goals and decision-making criteria. And we hope this afternoon to explore on how to overcome these challenges, and areas to further work will be proposed. With this short introduction, I would like to invite our first speaker, which is Monica Garcia Alonso. She is an independent consultant and director at Estel Consult. Monica has been working into the area of risk assessment of GM crops and regulatory affairs for over 20 years. And she will provide us an overview on how problem formulation can be used to construct fit-for-purpose environmental risk assessment of regulated stressors. Monica, the floor is yours.
Thank you very much for the introduction, and thank you very much for the invitation from the organizers to give this presentation. Unfortunately, I got the slot after lunch, which is never, the best ones, but I can assure you that the presentation I'm going to give you is actually quite simple, I hope that the concepts are clear and sets the scene for the presentations that will come afterwards that will give you more specific examples of the problem formulation. So what I would like to talk about is the basics of using the problem formulation to construct fit-for-purpose environmental risk assessments of regulated stressors. So as we all know, and this is something we've been discussing yesterday afternoon and this morning as well, we do risk assessments to evaluate the likelihood that adverse effects in the environment may occur as a result of a new stressor, let it be biological, physical, or chemical. And we have heard as well, and we agree that these environmental risk assessments need to be as fit for purpose as possible. So they need to focus on the key aspects that the assessment must consider, and also they need to provide relevant information for decision makers, because at the end of the day, what we are trying to do is facilitate decision making. So there was a discussion yesterday about what fit for purpose means. In my view, in the case of environmental risk assessments, this is kind of the definition that I like to apply, is something where we use the right tools to adequately answer the risk manager's questions. And we must ensure that the risk assessment has validity. So the assessment measures exactly what we are set out to measure, that it's got reliability, so it's consistent, it's accurate as possible, and as realistic as possible. And also transparency. So it does clearly explain the logic of what has been done, why it has been done, and how it has been done. So I guess that this links with the presentation that we saw yesterday about communication and making sure that, that is something that can easily be understood. So the situation in the EU, as we know, there are different kinds of stressors, and all of them seem to be under different regulations. So there are lots of guidances on how to conduct risk assessments, but they seem to vary depending on the risk, on the stressor that we are considering. And I just put here a couple of examples. We have, for example, the pesticide risk assessments that are under Regulation 1107/2009, and they seem to be very well defined. They are well codified. They have clear tiered approaches with some trigger values, and there's a clear path towards the risk assessment. When it comes to, for example, genetically modified crops, they are under another regulation, and in this case, what we found, especially at the beginning, it was not straightforward to apply everything that we knew about pesticides directly to GM crops. So we had to, look at other ways in which these risk assessments could be conducted. And one of the things that became quite clear from the start is that these risk assessments might be more case by case, in which, depending on the product, you might have different approaches. So there are some data requirements that are common to all, but the risk assessment will be tailored depending on which product you are assessing. And now we have these emerging technologies that are coming through, where, like the challenges at the moment to see whether we can apply these technologies or whether we need to do something different for the environmental risk assessment. So what we are after is always to ensure that whatever risk assessment we do is going to be fit for purpose. So one of the tools that has been identified as potentially useful for these risk assessments that are more open-ended is the problem formulation. And it's quite funny because any risk assessor that has trained as a risk assessor will use problem formulation implicitly. It's not something you think about, you just do it because that's the way the risk assessor mind works. But it's not always obvious when we are trying to apply risk assessment to new areas, to newcomers. They might not immediately see this differentiation or this methodology as such. So it was early described by the US EPA in the framework of pesticide risk assessment, and they explicitly explain what they mean by problem formulation. And these seem like a very good model to follow when we started looking at how to try to harmonize and to organize the environmental risk assessments for GM crops. So as a result of that, there were many publications, but also EFSA took this idea, and in the environmental risk assessment guidance for GMOs that was published in 2010, the problem formulation is there quite prominent, and it explains how to do the risk assessment, using these different steps in the problem formulation. And it's something that applicants have to follow when they submit a risk assessment for GMOs. Other countries are also taking this up. It's now in the regulation in India, for example. So it's something that is gathering momentum, and it's being identified as a useful tool. And we think that it might be also useful for new emerging technologies, in that it does help to focus and organize the risk assessment, and it might increase the chance that these risk assessments are fit for purpose. So these are some examples of the flurry of publications that came up regarding how to apply problem formulation to the risk assessment of genetically modified crops. These are only examples. There are more of these publications. So one question that is often asked is, well, what exactly is problem formulation. And if you look at the publications I just put up, the different steps might be described in different ways. So there's the details, might be different, but at the end of the day, it's just a method to actually help organizing the risk assessment and make it more transparent. So it allows the organization of the information in a logical way. It does consider the protection goals. It considers the relevant information that you need to assess the key questions that you have chosen for the risk assessment. It facilitates an initial risk characterization, so you can establish if the risk characterization can be completed at this step or whether you need more information. And if you need more information, it helps you to clearly establish what information that is. So you focus the search for the information for the risk assessment. So in plain words, I would say that problem formulation is really what is it that we are going to assess, what information do we already have for the assessment, do we have enough information, and if we don't, what information do we need? Just a method. So I'm going to go through the different components of the problem formulation as I see it. As I said, other publications might see different steps, but this is what we do usually for genetically modified crops. So we take into account the protection goals, then we gather the relevant information that we already have before we start producing more information. We do an initial risk characterization, and then we see if with that information we can reach a conclusion, and if not, then we plan what do we need to generate to actually be able to complete the risk assessment. And the whole of the steps is what constitutes the problem formulation. So I'm going to go through each of the steps. So protection goals, we've been discussing it before. The protection goals, we know that they're not always clear, and that in different regulatory frameworks they might differ, or maybe even in some countries, they might have a slightly preference for some protection goals or other. So one thing it is clear is that you need to be very clear and have a good understanding on what are the protection goals that you are dealing with when you're doing your risk assessment, so to ensure that it's fit for purpose. And usually these protection goals are set by policy. We've talked about this before as well, and are very broad and not always very clear. So when it comes to doing the risk assessment, we're going to have to translate them into more operational protection goals that will then facilitate formulating these testable hypotheses, for which then we can conduct the studies and corroborate the hypothesis. So again, there are a number of publications that have tried to address this and try to help in moving from policy protection goals into specific protection goals, assessment endpoints, and measurement endpoints. So I'm not going to go much into this because you will see examples, real examples of this in the presentations this afternoon. So the idea is that we go from very broad policy protection goals that are telling us what the country policy is to more operational protection goals that allow us to choose what aspects of that country policy we are going to include in our risk assessment, what assessment endpoints we are going to deal with in the risk assessment, so what will be assessed, and then what are the best measurement endpoints to find, to address these assessment endpoints. So what are we going to measure? So we go from something really broad into something quite specific. So in terms of gathering relevant information, this is fairly obvious for any risk assessor, but the thing to remember is that what we are doing is not just collect any information, but collect information that is going to help us choose information that is relevant to the test hypothesis that we have chosen and the assessment endpoints that we are going to address. And this can come from studies done during the regulatory packages, or peer-reviewed publications, or from previous risk assessments maybe done in other geographies. So any relevant information that can help us, we can add to this. And then we have this step of the initial risk characterization. And I call it initial because at this step we might just include, like information that we have from the data requirements. And before we go and undertake lots of new studies, we might want to take stock and look at what we have at this point. So we know that to do the risk assessment, this is a function of hazard and exposure. So most of the time we're going to look at hazard and exposure information. So in some cases in here, we'll be able to say, well, actually, there is no exposure for this particular group of organisms. And at this stage, without gathering much more data, we can actually conclude that the risk is going to be low. But if we don't have enough information on either hazard or exposure, certainly we have to go ahead and continue gathering information until we can reach this conclusion. So I'm going to put a very simple, very quick example. For Gmos, for example, one of the data requirements for all products is to provide an expression study where you show whether the protein is expressed in different plant parts. So you know that if it's expressed in the leaves, there might be exposure to herbivores. And if it's expressed in the roots, there might be exposure to soil organisms. If there is expression in the pollen, then pollinators might be affected. But if the expression study tells me that there's no expression of the protein in pollen, the exposure to pollinators will be very low. And at this stage, I can say, well, it's very unlikely that there's going to be a risk to pollinators. Something fairly simple that it might seem quite obvious for pesticide risk assessments, but for GMO risk assessments, in some cases, it's not so obvious. And, it's worth going through this pathway. And then it comes to the fact that we cannot always measure some of these things. So very often for this kind of assessments, we might need also to consider qualitative measures. And this is something, again, that is accepted in some risk assessment frameworks. And the key thing is to have something that is harmonized. So we have the key of what highly likely or likely means. And then we can all understand each other and reach the same conclusions. This is an example from the regulators in Australia. They published this in their framework. So this is something that is fairly commonly used. And then if we identify that we need more information, then usually that means that we have to go further into finding information on hazard or on exposure, but always taking into account that the information has to be relevant to the assessment endpoints we've chosen and to the hypotheses we've chosen. So how to actually do this? Implement this in practical terms. One of the tools that has been identified as useful is the use of these pathways to harm. And the pathways to harm, what they do is you take into consideration what is the proposed activity that you are considering, and then taking into account the protection goals, you define what harm you're trying to assess, what are your assessment endpoints. And in between, then you can list all the different steps that need to occur from this activity to the actual harm. So you have a series of events that must happen. And for each of these events, you can formulate a hypothesis that then you can test with information, either with a study or information that you have collected. So the hypotheses that you choose are going to be dependent on which definitions the harm you're working or in which decision-making criteria you're following. But for example, when it comes to the risk characterization, there are different scenarios. So you might find that the hypotheses are false or they are not tested or only weakly tested, so you don't have really much information on any of them. And then really the conclusion would be, well, you have either high risk or high uncertainty and you have to do some homework and then carry on with your risk assessment to resolve this. Or it could be that one of these hypotheses is corroborated and then you know that you can block the pathway and you can at this stage say, well, actually the risk will be negligible. In some occasions, we'll be more in the scenario of a weight of evidence approach, where not one of these hypotheses we know exactly the information or any of them, but we get bits of information from each of them, and altogether might allow us to make a risk conclusion as well. So that's the weight of evidence approach. So all this is included in the risk characterization. So I'm going to give you a very quick example, very simple as well, because you will see more examples of this throughout the afternoon. But for example, the cultivation of Bt maize, there is a concern that they might lead to a population decline of valued butterflies. I'm sure that you are aware of the monarch butterfly type of example. Here we can list the different events that have to happen for this to be realized. So the Bt protein has to be expressed in the pollen in the first place, that the Bt pollen has to reach the food plant of the caterpillar of the butterfly species that we are concerned about, that caterpillar actually eats the pollen, so it's there when the pollen is there, so they coincide spatially and temporally, and that the Bt protein has to be, at the end of the day, toxic. So we have the both aspects, the exposure and the toxicity. So for each of these steps, we can formulate a hypothesis. So for the first one, we can say there's no Bt protein in the pollen, and what we are trying to do is corroborate this hypothesis. Or for the second one, we can say there's no pollen on the food plant, or the pollen is not eaten because they don't coincide, or the pollen is not toxic. And sometimes we might find that this toxicity might be sublethal or something, and there is some effect, but it doesn't actually affect the population. So these are the different events, and we want to test, some of these hypotheses. So it could be the situation that we decide to test them all, but that might not be always practical, because if we see one of them for which we have information up front that help us with the risk characterization at this stage, we might be able to just block the pathway. And for example, as I said before, we always do an expression study. So if in an expression study you found that there was no expression in the pollen, then we can say, well, this step is not, this is corroborated, there's no Bt protein in the pollen. We can break the pathway in terms of no exposure, so we can break it at this stage, and that's it. So we can actually do an initial risk characterization with this existing data that we already have. Once you block the pathway in one point, then trying to block the pathway at every single point might not be necessary or practical. So you don't always undertake testing all the hypotheses. But there might be some cases in which you really don't have enough information at this stage to actually block the pathway, and then it's when you do your your analysis plan to actually see which studies you might want to do to break this. So in summary, problem formulation does take into consideration the protection goals. So there's a clear definition of what is being assessed and what is regarded as harmful. You gather relevant data so you can test the hypothesis that you have formulated. You do an initial risk characterization with the data you have, and then you see if you can reach a conclusion, and if you can, you have to generate more data. So in summary, the environmental risk assessments that we want to conduct are done to facilitate decision making, and they must be fit for purpose. The problem formulation might provide a useful method for environmental risk assessments that you could apply to any kind of stressor. And you can implement this using pathways to harm. So you can take into account these protection goals, you can formulate the hypothesis, you can make risk conclusions, and you can identify information that must be gathered to actually complete the risk assessment. So that's all. Thank you for your attention.
Thank you very much. Thank you very much, Monica, for this excellent overview how problem formulation can ensure that we will provide fit-for-purpose risk assessment which are valid, reliable, and transparent. Are there any questions from the audience? It's difficult to see on this channel. To Monica? I don't see none. Then can we have the app with the question from the app? There are none from the app? Okay, good. So Monica, you were so clear with this good overview. So then thank you very much, Monica. Okay. Now I would like to invite our second speaker, who is Jörg Romeis. He heads the Biosafety Research Group at Agroscope in Zurich, Switzerland. Agroscope is the Swiss Center of Excellence for Agricultural Research and is affiliated with the Federal Office for Agriculture, and he works for more than 17 years in risk assessment of GM crops, with specific emphasis on design and execution of non-target laboratory studies. Jörg will present to us the problem formulation for the environmental risk assessment of insecticidal RNA interference-based genetically modified plants and RNA-based pesticides and the effects of non-arthropod or non-target arthropods. Jörg, the floor is yours.
So the title was given to me, so it's extremely long, but I think you got it. So I thank Monica for the excellent introduction. I have my background in GM plant risk assessment, so if I say anything unsensible about the pesticide world, please correct me, and I'm happy to learn, but I try my... Where do I have to point this? Just one slide on RNA interference, RNAi. That's all that you have to know. So we talk about a new kind of mode of action to kill insects or to fight diseases. I will focus on dietary RNAi, so that means that the trigger, the double-stranded RNA, has to be ingested, as we have the case, with transgenic plants or with sprayable insecticides. RNAi is an endogenous sequence-specific silencing mechanism that is elicited by small RNA molecules. So the insects will ingest a double-stranded RNA. The double-stranded RNA, after ingestion, will enter the gut epithelial cells. Other cells in the body will be processed into small interfering RNAs, which are typically 21 to 25 base pairs in length. These small interfering RNAs will enter a complex with a molecule called RISC, and this RISC complex will then bind to, in a very sequence-specific manner, to messenger RNA in the organism, in the cell, and by doing that it will block the protein synthesis. So this is in very brief how this works. One slide also on problem formulation. Monica has elaborated in that in much more detail. This is very important as a first step in environmental risk assessment. We have to be clear about what is it that we want to protect. And in an ideal world, the protection goals would not be affected by the technology that we are regulating. So they should be generally applying to pesticides, to GM plants, and so on. In reality, they are not. Once we are clear about what we want to protect, we can think about how our activity, planting of a GM plant or using an RNAi spray, could harm this protection goal. And if we have done that, this helps us to develop risk hypotheses that we can then test in the following analysis. Now I will focus on arthropods, and arthropods play a very important role in our environment, and they contribute to a number of ecosystem services. Some are listed here, so they are of cultural service. They support cultural services. We have a lot of protected species. They are acting as pollinators, decomposers. I will focus on my area of expertise, that is biological control. So we're talking about predators, for example, ladybird beetles that eat or help to control insect pests in our crop systems. Now, if we are at that stage, Monica has shown that, and I fill this now with my example. If it's biological control that is provided by predators, if this is what we are concerned about, this is what should not be harmed, we can then on top write, this is our intervention, the growing of a double-stranded RNA expressing plant or the use of a double-stranded RNA containing pesticide. How could that lead to harm to biological control? Now, this is a very simplistic picture. We can go in much more detail, but what you see here is it's two pathways that I have sketched out. On the one hand, the compound could be present in the pollen. It could be that, or it could be picked up by herbivores, insects that feed on the plant. The predator could be ingesting this compound when it feeds on the pollen or when it feeds on the herbivores. If the compound is still biologically active at that point in time, it might cause an adverse effect in the predator. This adverse effect might be big enough to change the populations or reduce the populations of these predators, and this might lead to a disruption of biocontrol. And in principle, if we block this pathway at any given point with a very high certainty, we can be sure that our intervention does not lead to harm. Now, what we also see here, if we just prove that the compound of interest is not present in pollen, we only block one pathway, we don't block both. So it makes some sense to do tests at this lower end of the spectrum, because then we might block all the pathways above. And what you also see here, you see questions about exposure, but also questions about hazard. Now, if I think about an insecticide or a spray product that contains double-stranded RNA, of course, some of the pathways might be the same for our predator. It could be that the herbivores ingest this spray product. It might still be stable and affect our predator, but it might also be that the predators are present in the field, they are covered by the spray, they groom themselves, or they clean themselves, and by that mode they might ingest the active compound and be affected. So very simple pathways, but that helps us to ask questions that we can address in the risk assessment. And I have listed here simply a number of different questions that we could ask, and they are focusing on either exposure or on hazard. So you can, as we have said or heard before, you can check, is this molecule of interest present in the pollen? Is it present in the herbivore? Are the predators actually present in the field when you spray? So are they actually getting in contact with the material? Do the herbivores that feed on the plant or on the spray product, do they still contain biologically active double-stranded RNA, or is it degraded already? These are all great questions, but the question that we typically address for both pesticides as well as for insecticidal BT plants, for example, is the question about sensitivity in our organism. This is a question which comes pretty low in these pathways. So if we can show that insects are not sensitive to a particular stressor, we might not have to worry so much about the exposure here. So and that brings me to my favorite, risk hypotheses. It's similar use for pesticides as well as for GM plants or for BT plants, is that the double-stranded RNA does not affect our valued arthropods, our predators, at the concentration as it is in the field. And if we are at that point, I think we can start to think whether we have the data to address this hypothesis from earlier studies or whether we go into testing. Typically, when we test this hypothesis, we do that in a tiered risk assessment framework that is, again, similarly used in the pesticide world as in the GM world. You start with first-tier assays in the lab where you have a chance to expose your insects to worst-case exposure levels, very high doses, and you have very controlled conditions, and you can have a very high power in detecting adverse effects caused by this compound. If you don't see an effect there, it is very unlikely that this compound at much lower doses will have non-target effects in the field. Of course, if you see effects under these worst-case conditions, you continue with more and more realistic tests, eventually even field tests, to see whether there is actually adverse consequences occurring from this toxic effect. So we are all very familiar with this. What we now have to reconsider before we go into this testing is the mechanisms again. How could RNAi affect non-target species? And I have listed here the three major elements. The first one is what we call off-target effects. This is sequence depending. So that means that this one molecule that we have binds in a sequence-specific manner anywhere in the genome of our non-target insects and disrupts the protein synthesis. So it could be the gene that is equivalent to the target gene in our pest species, but it could also be any other gene in the genome. So we are talking about a very sequence-specific effect. The other two, the general immune stimulation and the saturation of the RNAi machinery, this is something that we call sequence-independent effects. There is some evidence that insects respond to high doses of RNA in a very unspecific manner. So they get into trouble, and this will have physiological consequences for the insects at high concentrations. But if I know all that, I think we are still fine if we do feeding studies as we have done in the past for example, Bt toxins, as we have done for systemic insecticides. But what I believe what we have to do or change is that we have to look at some different endpoints as in the past. So in the past, our testing has very much focused on mortality or death of the insects. We have also looked at some sublethal endpoints, but I think here, since we have these unspecific effects, also here the off-target effects, we don't really know where this molecule is binding in the genome. It's difficult to predict the non-target effects that could occur, and therefore I would urge us that we put more emphasis on sublethal endpoints and as a consequence also extend the duration of the tests. Now, if we have some idea about what we try to do, is the next question is what species are we actually testing with? So our protection goal is biological control, but which species are we looking at? If we look in the GM guidelines from EFSA for GM plant cultivation, we see that the system there is pretty flexible. So we talk about a case-by-case risk assessment approach. We can select the species that are used for our toxicological tests case by case according to these kind of three rough principles. First of all, we try to look at species or test species that are representatives of value taxa and functional groups that are out there in the field, also considering the species that are most likely to be exposed to the stressor, and there is always some practicability point to consider here. We have to have test systems available and species available to do the testing. But I think where it now becomes interesting is this middle part, because we can also choose species from which, based on our knowledge, we can predict or believe that they are most likely to be sensitive. In the Bt world, if you have a Bt toxin that is there to control beetle pests, it is simply more likely that beneficial beetles like ladybird beetles will be affected. So you put more emphasis there. Now here with the RNAi, I think we have some handle already to identify species that are most likely to be sensitive, and the literature in this field is growing massively day by day, but it appears that different arthropod orders differ in their sensitivity to dietary, so that means orally ingested double-stranded RNA. We know that beetles, coleopterans in particular, are very sensitive. That's why the first product that will be out probably next year in the field in the US is targeting a beetle pest. We know that lepidopterans, for example, are far less sensitive. We know that dipterans appear to be less sensitive. So we can use this knowledge to select our species. We know that phylogenetic, because everything is sequence specific, or the off-target effects are sequence dependent. There is good indications also in the literature that the phylogenetic relationship between our target species and the non-target species plays a role. And last but not least, we can use the tool of bioinformatics, because we can use genome information, if we have it, to predict where binding occurs in our non-target species and where testing makes most sense. And this field, as I said, is growing at a steady path. Now, in the pesticide world, the whole thing looks a little bit different. And if I look in the papers, in the guidelines, I see that the system is much less flexible. A lot of things are defined. This goes down to species that have to be tested, to principles according to which the tests have to be conducted. Even the test parameters, the endpoints are fixed. And I think this brings us a little bit into difficulties here, because if we look for non-target species, and I know the system is a bit more flexible than I show it here, but I couldn't find, a document which tells me, where the flexibility comes from. Because if I look at all the regulatory documents, it tells me you have to test these species. And if we go down to the terrestrial arthropods, it's the honeybee and it's these two natural enemies. So that makes a lot of sense for the classical insecticides. So people have thought a lot about this, but it doesn't make sense for the RNAi, for these new molecules. What, to my surprise, when I was preparing this talk, I found a document which I find very intriguing. It's a scientific opinion from the PPR panel from EFSA from 2015, because there they have not talked about RNAi products that will show up, but they have talked about systemic insecticides a lot. And some conclusions you see here from this report, what they recognize is that we don't account for this oral exposure pathway well enough, which is the case already for systemic insecticides. We have problems to relate the endpoints that we are collecting today from a glass plate type of toxicity study to realistic exposures. If we think about systemic insecticides, we don't have standardized tests for testing oral exposure. And we need more tests that take into account these different routes of exposure, but also the different modes of actions. And I think this fits one-to-one to the double-stranded RNA discussion. So in conclusion, I think, I showed you, Monica did a much better job that this problem formulation, think what you are concerned about, think how this could be harmed, helps us to identify risk hypotheses that we can test. For the GM world, with all its flexibility, I think we are well set to assess the risks, non-target risks of double-stranded RNA expressing plants. The case-by-case approach allows us to select the most appropriate species. We have test systems available for the BT plants. We use it all the time globally. I think the amendment I would make is to put some more focus on sublethal endpoints for the reasons I elaborated. And I think for the spray products, I see some problems with the restrictions that we have to now. I think we have to open that up and get more flexibility to test species that are simply making more sense. And I would also like to remark here is because the pesticide people criticize that there is a lack of toxicities or test systems for oral exposure. I think for the GM world, we have those tests. So I think this is a great opportunity to bring these different units together and to learn from each other. And with this, I would like to thank you very much.
The mic is not, oh, mine is on now.
Now?
Oh, yes.
Okay. Elizabeth from GMO unit at EFSA. I have two questions actually. One is from your perspective, how useful is the bioinformatics at that point in time to get information on the broad range of non-target organisms for off-target effects? And the other one is, you mentioned sublethal endpoints should be measured instead of the more conventionally measured one, or in addition to mortality. Why do you think this is particularly important for the RNAi-based applications and not of general relevance?
Okay, two questions. So let's start with the endpoints first. I think, in a BT world, we have the expectation that this toxin is killing your target. So the first thing to expect in a non-target species is a toxic effect. And that's why it makes a lot of sense to look for mortality. In practice, people are also doing other measurements. So now here the problem is that we don't really know what the effect could be, because the sequence depending binding of the RNA could be anywhere in the genome. So it could affect any other gene, but not the gene that we are targeting in our pest species. And also these other effects that I mentioned, the general immune response and things like that, I think this will be very specific, and we don't really know how that would show up in an insect bioassay. And that's why I think, and I mean, we have this one, so far we have only one example, that brings me also to your second question. There is one event that has been approved by EPA last year. It's a corn rootworm event developed by Monsanto, which will be in the field next year. They already by themselves, they have decided to expand the duration of these feeding assays and to look more at sublethal effects, I think for exactly the same reason. It was not explicit in the documents that I have seen, but for the same reason. And what they have done also, that brings me to, the bioinformatics at this stage in time. I don't think that bioinformatics, it will assist, but it will not replace any other effect. It will assist, for example, and if the knowledge that we have on the genome of insects, and this is constantly growing, we might at some point use bioinformatic information to do a risk assessment. I wouldn't rule this out, but this is probably beyond my time horizon here. But we don't know. But I think now, and again, this American case has shown that they have used bioinformatics in addition to the feeding assays to show how specific these bindings really are. So they could show, besides all these feeding assays that they have done, that the activity of the double-stranded RNA molecule is really just within the same subfamily of the target insect. So you can use this as an additional information, but not replacing the results from the feeding assays at this point in time. We will see.
We will see. Are there any more questions from the audience, perhaps? No? Oh, yes, right at the back. You're waving at me? Oh, the app, right. So yes, we have a question here. Protection goals should be defined by risk managers. Why should risk managers set the same protection goals for all technologies? Societal benefits, values, and concerns may be very different. Could you perhaps comment on this?
Yeah, this is a nice one. I like that. But if I'm an ecologist, so if I think about nature and I'm concerned about ladybird beetles or honeybees is a better example, I don't like to see them killed, but I would not make a difference if they are killed by a pesticide or by a truck or by a GM plant, but in reality now we appear to put different levels of concerns or adapt or identify different protection goals for different technologies, but it's the same nature that we should protect. And I think it's just sensible to protect it from all stressors in kind of the same way. It's the harm that I'm coming from the harm, and this harm that I want to avoid. If I think about honeybees, I want to avoid from all technologies to happen.
Yeah, I think this touches very much on the societal perception of different types of risk from different technologies, which links to the discussions we had yesterday. Are there any more questions? If not, then I thank you, Jörg, very much for your talk. And I have the pleasure of inviting Dr. Alessio Ippolito. Alessio works for the pesticides unit here at EFSA, and he will give us a presentation on the environmental risk assessment of neonicotinoid insecticides for bees, a retrospective analysis of the problem formulation.
Thank you for the very nice introduction, and good afternoon, everyone. Indeed, this will be a retrospective analysis, so the focus will be probably slightly different than the other talk from this afternoon. But sometimes it's good to see what's back. Let's say what we have what we've seen, what we have done in the past before we move forward. So let me start by saying that the risk assessment of pesticides in the area of the environmental risk assessment of pesticide, the problem formulation is defined once for all in the relevance guidance documents. That was already nicely shown by Monica before. One might agree or disagree with it, but this is the status quo of the situation. Now, the start of our story is probably in the middle of 2000, where there were some incidents linked to the use of the neonicotinoids insecticides, and those incidents basically led regulators to think about what the problem formulation was at that time, and they basically led to the idea that that problem formulation needed to be updated. And the discussion that followed culminated in the publication of an opinion of EFSA in 2012, and finally in EFSA guidance document for bees in 2013. Since then, at EFSA, we have gained quite some experience in using that novel problem formulation and that novel risk assessment scheme. And at this point, we probably have sufficient experience to have a look backward and see, have a critical, let's say, assessment of the problem formulation. And indeed, this presentation is performing a retrospective analysis of the problem formulation within the EFSA guidance documents, using especially the latest assessment of the neonicotinoids as main reference point, let's say. So this is very shortly basically what I've already said, just to show you that this is a timeline of the different regulatory documents for the risk assessment on bees in Europe. As you can see, the last one are the one from EFSA, and from that moment on, there were quite a lot of assessments performed by EFSA, so that is the experience we built up during the last years. To perform this analysis, I asked myself, what are the characteristics of a good problem formulation? I haven't done such a good job as Monica did, but okay, I just came up with at least 3 very, very clear criteria that one can use as key for assessing it. And this is that a good problem formulation should provide clearly defined goals. It should make use of unambiguous endpoints, specifically it should avoid those that cannot be easily measured, and it should also, of course, able to identify all the relevant risk. Now, I'm not going to give you a full overview of the problem formulation as it is in the EFSA guidance document, because that will require the entire afternoon, but I will only focus on specific aspects, let's say, that I consider to be quite important because of the elements of novelty that was brought by the problem formulation in comparison to the previously existing one. And these are mainly the identification of the relevant route of exposure, the quantification of such an exposure, quantification of the effects, and then the consideration of spatial and temporal variability. Now, regarding the first point, the identification of the relevant route of exposure, I'm not going to speak about the exposure via contaminated pollen and nectar, because that is, passing the term, pretty obvious, and it was already, let's say, considered in the previously available risk assessment scheme and problem formulation. But I will definitely speak about dust drift. So dust drift is a relevant route of exposure when, especially for seed treatment and granules formulation. Basically what happens is when you drill the seed or when you broadcast the granules, they have dust that is lifted up from the field and transported, drifting out onto the vegetation off crop, and there the bees can be exposed. The relevance of this route of exposure once again became relevant in mid-2000. Again, the references to those incidents I was talking about before. And none of the previously available scheme actually proposed, a complete scheme for assessing the risk due to this route of exposure, while this guidance document does. On the other hand, the quantification of this exposure route is really complex. It depends on a lot of factors, and therefore current estimation also have some uncertainty, also because the amount of data it is based on is not so big. It has to be highlighted as well that the current scheme for refining these exposure values, let's say, has been proven somehow unpractical because, again, it requires really a lot of information which is difficult to put together, especially when this assessment is performed at the EU level. Another route of exposure that was introduced, let's say, somehow in a more organic scheme by the EFSA guidance document is guttation. And it's absolutely clear from the experience we've gained that this exposure route is important for single honeybees, or can be important for single honeybees, but its relevance at the higher level of organization, for example, at colony level, then it is more debatable for most crops. So that's something we should really look into with more detail, let's say. There's also a quite poor understanding of the mechanism behind the guttation. So the substance is basically taken up via the roots and then it's transported up to the plant, and then it's available in this water that is leaving the plant. But it's, at the moment, the assessment we made is really just a screening performed on water solubility, which of course is very rough, let's say. And once again, the current scheme for refining this has been proven unpractical due to the amount of information that one needs to collect. we know anyway that the route of exposure we consider are somehow, let's say, sufficient for the honeybees and all the bees that have similar bioecological traits. We know that there might be other route of exposure that are more relevant for other bee species because bee diversity in Europe is still quite high. However, we know this route of exposure exists or may exist, but we don't really know whether the one that we are currently assessing are protective or not for the one we are not really assessing at the moment. But there are activities anyway going on this issue, so we hope we will have more details in the future. Now, moving on to the quantification of the exposure, I think this is probably the part where the EFSA guidance document has made the biggest advancement, let's say. Unlike in the previous schemes, the exposure is quantifying unit the same dimension that it is also used for quantifying effects. And this is extremely important actually because it allows us to reduce the uncertainty related to complicated calibration exercise. So that is really important. And also what is really good is that we have an explicit consideration of the factors. There are multiple factors that are influencing exposure. So it's the residue levels, the content of sugar in the nectar, the consumption, etc., etc. And most of these parameters can actually be measured so that the exposure can actually be refined. And probably the most important thing is that for the first time we have what we call an exposure assessment goal. So we know what is the level of exposure specifically to a certain use that we need to achieve when we are assessing a specific effect. Now, for the quantification of the effect, we have to, let's say, split a little bit of discussion. And again, I think we can split by saying that for the main effects assessed, which is probably colony strength and the population abundance somehow, there are three thresholds of effects which are also specified and directly linked to the specific protection goal. So that is, of course, very good. What we measure at the lower tier for honeybees, but also for the other bees, is basically lethal effects, what we assess in the lab. And for that, at least for honeybees, we do have some kind of explicit link, which was done through modeling. So I think this ties pretty well with what Valerie was saying this morning. It is true that the model it was used is fairly simple, but anyway, I think the link is there and it is addressed. The specific protection goal for honeybees also include forager mortality, but this I have to say when it comes up to higher tiers, so to field and a semi-field study, that is fairly difficult to measure. So that's also something that should be probably better addressed. For other bees, the situation is slightly different because the influence of the lethal effects that are measured at the lower tier is less, let's say, linked to the effect that you might observe at the higher level of biological organization. So again, colony or population. So that's something that can be improved. We are, and this is probably connected to what Jörg was presenting before, we are already recommending measuring a range of sublethal parameters at the higher tier, but also at the lower tier. But when we actually did performing the risk assessment for the neonicotinoids, we had a lot of sublethal data, but we realized it was really difficult to use that information for concluding on the risk to address the specific protection goal because that linkage was actually missing. Another topic that I would like to talk, because I think it's really important, is that bees in general live in a variable environment, so we need to account for variability in both the exposure and the effects. For what concerns the exposure, I think the spatial variability is quite well addressed in the problem formulation as it is in the EFSA guidance document, and it is actually the theoretical basis for the exposure assessment goal I was mentioning before. So that is pretty well covered, I would say. For what concerns the effect, it's slightly different, I would say, because we assume that effects are different in different parts, let's say, of the area of interest, but it's mainly driven by the difference in the exposure. We don't explicitly account for other factors that might influence, let's say, the effects. For what concerns the temporal variability, once again, the exposure is pretty well addressed, I would say, because we do consider change in the exposure due to time, especially for spray application, as we consider the decline in time of the active substance due to the processes that are intervening. It is less relevant for systemic insecticide because these are translocation from the soil or the lower part of the plants to the upper part, and that gives a more constant exposure. For the effects, again, we have to split the discussion in two main components of the specific protection goal. For what concerns forager mortality, which is really applicable only for honeybees, there is an explicit consideration of the time in the assessment of the effects. While for what concerns the specific protection goal for colony strength and population abundance, there is not really an explicit consideration of time, and that is also quite problematic when you consider that these parameters are actually quite variable in time, and therefore to really, let's say, assess an effect, not having a definition of a time frame is quite complicated. So just to conclude, we have performed an extensive literature research during the last assessment of the neonicotinoid substance. We've been looking at, if I'm not wrong, 1,600 different studies. And I think we can say that at least for honeybees, we can be sure that the relevant route of exposure are considered in the problem formulation as it is now. We might debate on the fact that some of them, particularly glutation, is actually relevant at the higher level of organization, but certainly, well, almost certainly for the information we have available, we haven't missed relevant route of exposure for honeybees. In my opinion, the exposure characterization has made a huge advancement, at least in conceptual terms, compared to the previously available schemes. Also, the refinement strategy which is proposed in the guidance document is logic and very consistent. For pollen and nectar, it is also quite straightforward, I would say, but for other route of exposure, as glutation and dust, it is slightly more impractical. And to conclude on the exposure, I believe that the spatial and temporal variability is quite well addressed. When it comes to the effect, we can say that the problem formulation resulted indeed in a consistent scheme across all the tiers. I haven't had the time to really look into the single tier of the assessment, but that's what it is. And for the endpoints that have direct link with the effect, let's say, mentioned in the SPG, we have really a good match, and that allows us to perform quite informed, let's say, assessment. Well, for the use of the other endpoints, specifically sublethal endpoints, which are considered relevant by the guidance document, then the assessment is more problematic because of that missing link with the SPG. And again, the spatial temporal variation of the effect, mainly in terms of measured endpoints, needs to be better addressed, in my opinion, at the moment. So with that, I conclude, and well, thank you for your attention.
Thank you very much, Alessio. I think this was also another really nice example, the retrospective look at the problem formulation for neonicotinoids. So I first open up to you, the audience, and I see already an eager question at the front. Please.
Thank you. So in your presentation, and I'm pretty sure the other two so far this afternoon, my impression is that the problem formulation, the way you're describing it, is including effects assessment and exposure assessment and the risk characterization. And I'm more familiar with thinking about problem formulation as step one, and then you have the exposure assessment and the effects assessment as separate steps, which you then bring together in the risk characterization, which I think is consistent with the US EPA's framework, which several of you cited. So now I'm really confused. How are you all thinking? What's included in the problem formulation step?
Well, I have to say, personally, before this presentation, I never really thought of problem formulation in a formal way, let's say. So this was really just my first attempt with that. But the impression I had while also talking when organizing this session was really that problem formulation, as you were implying, as it is conceived, let's say, in this session, is really a process that encompasses all the different steps of the assessment. So not really just a preliminary phase, but also how you would design the assessment and how you will assess all the different aspects of the risk assessment throughout the different tiers as well. So that was my interpretation of it, and I see that is also quite consistent with the other presentation from this afternoon.
Did you want to add something there, Monica, perhaps? Might we have a micro? Voila, here we are. Have this one.
Yeah, and
Oh, it's not on. Do you perhaps have another microphone?
hold on. Yeah, you can hear me? Yeah.
Oh, it works, thanks.
I'm not surprised you're confused because I have noticed, and I did mention that different problem formulation schemes have like different definitions of the different steps, but consistently what we use, as you have seen from Jörg and all that, is I think that the key difference is that this initial, and I call it initial risk characterization, is the one that you do with the information you already have before you start undertaking studies to find the information that you need and all that. So it's the very first step, yes, but using information that you already have. But then you use it again to generate the rest of the information. So it's an iterative process. But you would do an initial risk characterization from the very beginning, just to take stock of what you already have, and then identify what's missing, and then carry on and do. I don't know if Jörg would agree with that.
Thank you for that clarification. Were there any more questions from the floor? Or if not, should we see our app? Whether our app brings us any questions? No questions from the app. Okay. I think then, thank you again, Alessio. We would conclude. And then I have the pleasure to invite the next speaker, Dr. Klaus Svensson. Thank you, Klaus. Klaus comes from NERC, the Centre for Ecology and Hydrology in the UK, and will present to us current scientific developments in the environmental risk assessment of nanomaterials. Thank you.
Thank you very much. I'd like to, first of all, thank the organisers for inviting me to come here. I'm going to take a slightly different tack. I'm going to look forward to what problems nano might have in store for us, why it might be special, and the things we have to think about when we want to make our problem formulations and select our protection goals. Again, the title was given to me, but it's nice and broad, so I will go around the topic as much as I can. Now, I'll try and cover an introduction of what nano is, for those who doesn't know, where it is, it's in surprisingly many places, and then look at the fundamentals of why the risk assessment, and specifically environmental risk assessment of nanoforms, is different to that of classical chemicals. Then I'll go through how nanomaterials might benefit and impact the things we value. Am I too close? Okay. Thank you very much. And both look at direct application examples and also on unintended exposures and how some people might have to think about their risk assessment, also including an error element to it, even when they think they're finished. Then I'll finish off with the implications for the error, considerations, approaches and tools, are there new hazards, exposure routes, how environmental fate becomes really important, whether the biodegradation, accumulation, biomagnification are different, and a short bit on the technical and analytical needs and what our test guidances are. So what is nano and where is it? Well, it's everywhere. The main thing is its engineering of materials at the very small scale. The easiest example is suntan lotion, where you all know the classical suntan lotion on the right here, the nice white smeared stuff. When you make it nano, you can take it down to below 80 nanometers and get it below the wavelength of normal light. which means you're still blocking the UV light and absorbing the UV light, but you're not interacting with the visible light, so you have a nice appearance on the beach while still being safe. The other place in the market is everywhere. I think is the easiest way to describe it. There's materials, there's structural and functional materials, anywhere in personal care products, water filters, so on and so forth, but also on the market for use in agricultural purposes. So in terms of EFSA, where are the main places for nano? This is a review from 2016, which goes through the different parts of EFSA, if you want, where nanomaterials have been seen applied. You can see there's a lot in food additives. This is one of the places where I think it might leave the organism that you're worried about, but there's a protection goal further down the line where it enters the environment after that. We'll get to that. You'll see below that there's all sorts of materials involved here, from a lot of titanium dioxide in food additives, but also, surprisingly maybe to some of you, a lot of silver being used in quite a few of the categories: feed additives, biocides and food contact materials, but also food additives. I'll get back to silver later. Now, the difference and what it defines nanomaterials, I have to say the EC short definition, because this definition has been short, it's become longer, it's become shorter, it's now longer again. But the basic part of it is it's a material where 50% or more of the particles present in your material, by number, has a size that has one or more dimensions less than 100 nanometers. Now, what's 100 nanometers? If we go from a thumb and we go down to a cell, which takes us 3 steps here, then we've gone from 20 millimeters to 20 micrometers. If we take another 3 steps, then we get down to 20 nanometers. So we're down in the size of histones, protein, subunits. For the very small nanomaterials, we're into things that interact actually in the matrix of folded proteins. So in terms of what that means for your risk assessment, it's kind of like being asked to do the risk assessment for a PM10, but most of your data is on small stones, because it's the same scale factor between a small stone of 20 millimeters and a PM10 that we have from PM10, the ultrafines, down to the nanoscale. So it's a physically different world. If we look at what the main differences are for the environmental risk assessment between classical chemicals and nanoforms, classical chemicals generally behave in a dissolved manner in the environment, whereas nano is suspended materials, and they behave differently. So if we look at ions, they generally move around, they go on and off surfaces, and we generally consider having an equilibrium after some time. Particles will, when they bump into stuff, bind, and they will stay there. How fast they bind depends on their affinity for the solid matter around, whether that's cells or parts of the environment. So you end up with no equilibrium, and you basically have a kinetic fate descriptor for your particles. So instead of it settling down to having a traditional equilibrium at chemical concentration, you end up with these situations where the small particles bind to larger entities, have co-transport, and it becomes about fluxes, and you'll see the build-up on the solid phases carrying on in time. So where's nanotechnology for EFSA? It's basically in all these places, and EFSA has already started producing guidance on some of the elements of it in terms of food and feed. There's more to come and more is needed. I'll try and cut out some examples from plant protection and from chemical contamination just to illustrate some of the points I've been trying to make. If we look at plant disease and nutrition, nanoscale nutrients have been used to deal with root disease, but not in the way you might think about. So fungal pathogens reduce crop yield by 20%, roughly worth 200 million in the US, despite 600 million being used to control it. Many micronutrients are either stimulating or an intrinsic part of the plant defense system, and these nutrients are very limited in availability once they're in soils and roots struggle to extract them. So delivering more into the soil, you're still fighting an uphill battle. So the question here was, can you add them topically via the leaf and have them translocated to the root and help the plant defend itself in that manner? And this is work from Jason White's lab in Connecticut. So they basically took nanoscale micronutrients, applied them to the leaf in greenhouses where the plants were safe from the infestations in the soil. Then once the plants were grown and had the nanomaterials applied or various controls, they transplanted them into infested soils in the field and then monitored the yield, disease and root content of the material of the micronutrients. So the single foliar application was in the lab, in the greenhouse, but once in the field they observed an increased yield in the plants that had the copper oxide treatment applied. They had greater disease suppression. So you can see here that the nano form has the lowest area under the disease progression curve, and you can see the yield being higher than in most of the other cases. You can also see that in the root the copper is increased, although it's only applied topically on the plants. So for a small application on the plant, so where you wanted it, not putting loads in the soil, they calculated that for $44 an acre, they could increase the yield about 30% in value. So this is obviously not killing the disease outright, it's helping the plant itself, but it has benefits that you have to consider both in terms of what the alternative would have been to try and combat these fungicide infections, but also in terms of applying what you're trying to do in exactly the place you want it. So release and exposure nanomaterial fate in the environment, this is really where it gets different. This is the project I coordinate, Nanophase, and this is where we are trying to bring together a lot of work from past EU projects. I'll try and go through it reasonably slowly, but it basically comes down to, like for any product, you have a synthesis of the active. So here these are particles. They get put into project manufacturing in various ways. They get distributed, they get used. Some gets recycled, most gets waste managed somehow. At all these stages, particles can leave and enter various parts of the environment, either the air, the soil, or the water, through different routes. But the problem here is that while for most other chemicals, the form that leaves is the same, for the nanomaterials, they transform at each of these steps. So getting this material and testing the hazard for it might not mean very much in terms of what the hazard of what is actually entering the environment is. Once we're in the environment, you have these cell reactors, environmental cell reactors we call them, and basically the transformation continues. It's not degradation as we consider it normally. They will transform chemically and acquire, most of the time, less reactivity, but other things do happen. So we have to take care of these when we make our problem formulation, because what we're trying to assess isn't necessarily what is the exposure relevant form. We then have to try and work out that because it behaves as a particle rather than as a solute, the fate might make it travel differently, so we end up with hotspots in places where we wouldn't normally expect them. So this is what we're having to try and model based on understanding these processes. And that obviously comes with overlaying layers of population and industry locations, environmental chemistries, etc., etc. So if we take one example here where we have a nanomaterial that's made, whether it goes through a food additive route and gets excreted and into the wastewater stream, or it's industrial wastewater, doesn't really matter. It'll enter in an industrial waste water treatment plant, and one of the exit routes from there is the sludge product, which gets applied to soil, mainly agricultural soils, which then has obvious protection goals in terms of both the organisms that live there and the crops we grow there. If we try and look at what would happen if we take a standard test, and we only use pristine materials so far, we'll take one step at a time, and compare that to the field exposure. So this is a 4-week OECD standard earthworm 222, OECD 222 test, reproduction of earthworms. We look at how many juveniles are produced compared to or against the concentration in the soil. You have the soils being prepared and tested straight away. The soil's left for 2 months, 7 months, or 12 months before the test is conducted. You can see that the toxicity isn't great at the start, where we have no time progression. So the nanomaterial appears to have an EC50 of 1400 milligrams per kilogram, where silver has an EC50 of 50. So nanomaterials are a lot less toxic than the silver they're made from, which is a good answer if you're industry. However, if you let the soils age for 12 months, you get these data, so the bottom curve, where you find the nanomaterial is now down in the order of the same toxicity as the iron was to start with, and the ions have aged a bit like we would expect them to and become less toxic. So the standard test might not give you the worst case scenario. Now that's a fundamental problem, because we have designed all these tests to make sure when we put the chemical in, we get the worst case exposure we could have. That's how the hazard estimate works. Now, we also have to worry about testing the pristine versus the exposure relevant nanomaterials. So when silver goes through its sewage works, it'll go through and turn into silver sulfide. I'll have those slides at the end, behind the end, if anybody wants to see them. But basically, we do uptake experiments where you first have the worms, these are again earthworms, in soil that have silver in them for 24 hours, and then we take them out and put them in clean soil and see if they can excrete the material again. So this is pristine nanomaterials, and you can see the total silver measured by ICP-MS goes up and comes down. But when we extract the particles from the worms and measure them by single particle ICP-MS, we can see that only 15% or less of the silver in the organism is there as particulate silver. So they've dissolved, or been taken up, or been taken up and dissolved. Now, if you compare to the silver sulfide, that's going to be the real exposure in the environment, we have nearly no uptake, 10 times less uptake. But the uptake that there is, nearly 40% or more of it is in particulate form. Hard to see, but it was more this difference that I wanted to illustrate. So the two forms travel differently, they behave differently in the organisms. It's not much use to have the data for this if this is what your exposure problem formulation says should be out there. Similarly for plants, if we go through that, you can see we have 3 different soils of different pH and organic matter. So pH 7 through to 5.5, and organic matter from 2 down up to about 15. You can see that the soil concentration was 10, and the wheat accumulates that in 2 of the soils for pristine silver. If you use the silver sulfide, the accumulation is slightly less. Now then there's this third soil where they accumulate a whole lot more. Unfortunately, the standard soil is this one, so because this is fake chemistry being really important, maybe we have problems with the standard material, standard soils and media that we have. We have to consider the chemistries of these materials and whether there's other things we should be including to get the right data. If we go through the implications of what that means for error considerations, approaches and tools, you can see for the other new hazards, there's not really new hazards apart from a few ones of the suntan lotions creating photoreactivity and ROS generation. The routes are probably not new, but the rates and internal transport speeds and routes might be different, but the actual uptake routes are probably the same. There are things that obviously you have to consider with materials not being taken up as ions. But the exposure routes that we have to consider in the risk assessment are the same. Environmental fate is very different. It really rules what might be the possible nano-exposures, and it should inform what the forms are that we want to have as the exposure-relevant forms which the risk managers should consider. The timescales are long, much longer than they are for chemicals, which means current standard hazard tests may not present worst case, as I said. The easiest way to deal with this would be to pre-age the test media rather than run the long-term test. It would also be the cheaper option, probably also the more reproducible option. Biodegradation, accumulation, biomagnification. We need to test the relevant forms, so the difference between the silver and the silver sulfide. We also need to test the form, size and speciation of the internalised material so we know what's in there, because that's important for the next step of the risk assessment, who eats the product at the end. Technical analytical needs and test guidance. New analytical and testing techniques are needed. We need to try and keep them simple. There's a tendency for people to get really excited about their technologies and get to infinite detail. But really some of the answers we need are really simple, and we just need techniques that could be in all labs and break these simple answers down in a reproducible manner. We have to move from solute-based to kinetic tests. So some of these OECD tests that we're used to dealing with in ERA doesn't really work. We have to make them work on a kinetic basis. That works ongoing. There's techniques that we need to learn to understand, X-ray-based methods for speciation assessments. All of this is going on. There's a lot of work this autumn, both in ECHA and the OECD. We were in Paris with the OECD last week to try and deal with some of this. So now's the time to get your things on the list if you need to worry about it. So overall conclusions from those nanospecific elements is they will differ between the intended uses that you have, so you have to be careful when you do your problem formulation to include these nanospecific elements, think about them. Because the use is widespread both within agriculture and the benefits are many, we need to consider that the exposure in food supply and to the food supply could become significant. We need to understand the fate processes mechanism, both of the actions and the interactions, in order to be able to do accurate errors. And the nanotechnology has the potential to improve the agriculture, which means we must try and remember the trade-off between we can cure the problem one way. If we don't do that because there's some risk with it, what would the alternative be? Is that a worse alternative? This is slightly more soft again, but I think it's important in these new technologies. So we're learning, but if we get good guidance and direction, we can get through the rough stuff and get to where we want to be. And I guess where I want to be now is questions. Thank you very much.
Thank you, Klaus, for these useful insights on the current developments in nanotech testing for ERA. Can I see whether we have any questions from the audience?
There's one over here.
Here, please.
It's a bit hard to see up here.
No, the microphone is coming.
A question. I'm from EFSA, from Plant Health Unit, so I don't work with pesticide application, but I see that nanomaterial, micronutrients can be used both for fertilizer and for pesticides. And same for these two types of products. I mean, the same nanomaterial could be used in two different applications.
Yeah. I think it comes down, I think the risk assessment will be slightly different. For the fertilizers, I think if I was the producer, I would suggest that I can do more benefit applying less to the environment and applying it in places where it needs to be. So the off-site loadings would be less. For the pesticides, the most use of nanomaterials and pesticides is for exposure modification, which also means you can hang the pesticide on the leaf through the rain so it stays where it needs to be. But really the nano element of it comes down to will that exposure go elsewhere. The risk in it is still the active ingredient. I mean, the toxicities of the pesticides are orders of magnitude different to the toxicities of the nano element of that delivery mechanism. So I think the bit that will be new for both of them is you have to consider the fate much more. But for the nutrients, I think the fate will be mainly on the benefit side, and for the, which it also will for the pesticide, but there might be things you have to consider in terms of things that might become exposed in these nano-encaged pesticides that you wouldn't have seen otherwise. But I think it comes down to really considering the fate to make sure you're not missing things when you set up the problem formulation. Did that answer your question? Sort of. Okay. Well, we'll do it over a cup of coffee then. Anybody else?
There's still time for, thank you, there's still time for one additional question from the audience. If not, are there any questions from the app? No?
No?
Okay, so then we are right in time for the coffee break. Thank you very much, Klaus.
Thank you.
And I would like to ask you to be punctually back at 13:45 because we still have 4 speakers to go and the podium discussion. Enjoy your coffee.